think we're going to go ahead and get started. All right. So welcome everyone. It's been a while since we last met, and I'm just really excited to be spending time here again with all of you and with Dr. Jasleen Jolly. We're super excited to have everyone join us. It's a larger group, so I think there are going to be more people that will be joining us. We are recording it, and the recording will be on the cloud, so you can look at it again. Dr. Jolly's contact information will be also listed here when we send out the recording. So you can feel free to send any comments or questions to her individually. As always, we always like to thank our sponsor, that would be Genentech, for sponsoring and funding these webinars. And also just to tell you that if you have questions during our time together, go ahead and write them in the chat feature, and I'll share them with Dr. Jolly as she's presenting her information. So with all of that, I just want to say again, we're so excited to have you with us today. Thank you. And Dr. Jolly is the recipient of the 2021 Ricketts Family Research Award. So that's quite a notable thing, and we're very excited that she is on our team. And we're going to learn from her today about her work that she's doing with her team over at Oxford. You know what? You are still muted, Dr. Jolly.
My internet just cut out then, so I missed the end of that.
You're on.
Am I good to go? Okay, great. I'll just share my screen. I am so delighted to be here after working with Choroideremia for so long, and I see some familiar names in the audience. So during the Q&A session, please feel free to switch on your cameras and interact with me, because it would be lovely to see some faces as well. So I have moved from Oxford to Cambridge a few months ago, and I'll be continuing a lot of this work. So although I'm not employed by Oxford anymore, I've included Oxford in my affiliations just because a lot of the work I'm going to be talking about was originally done in Oxford, but will be continued in my new institution in the Vision and Eye Research Institute at Anglia, Ruskin. So I'll feel free to jump in with any questions as we go along or at the end. So just to go through what the research pathway is, the initial stages are the preclinical work. So this is the work that goes on in the lab. It's where you have to look at the animal models of the disease, come up with the therapy, try it out in the lab, and that before you can translate it into human trials. Then it goes into clinical trials, and you have phase 1, 2, and 3 trials typically. And then there are phase 4 and 5 trials, but they can happen post treatment being agreed upon. The phase 1, 2, and 3 are the essential stages. So phase 1 is the safety trial, and phase 2 is when you start looking at efficacy, and then phase 3 has to be a multi-center, multinational trial where you're using the final end product. So what would be the off-the-shelf product that would be employed if it were a treatment, because you want to make sure that whatever you're using would be viable when it's deployed, and it would be viable in a number of, in this case, surgeons' hands. So that's how the trials works. Then you have to get submit all of that information along with all the other studies, like the health economic studies, which looks at the cost benefit analysis, and the quality of life information studies that you've done. All of that information goes to the regulatory authorities, and then they decide whether they're going to approve it as a treatment. Then it goes to each individual country. So in the UK, it's the NICE, the National Institute of Clinical Excellence, who then decide whether the body, the NHS, so our National Health Service, is going to fund it. So in the US, it would be each of your insurance companies that has that negotiation, and then they agree the funding of it. So all of that is quite a long process. But from working on the trials and working within that process, I've realized that one key step is actually missed out. And this is what I want to focus on today. And that is outcome measures. And I'm going to show you why it's so important and why we need to be paying more attention to it. And I'm going to share with you why this has a direct relevance for the choroideremia journey, and why it affects what has been happening on the journey for you guys as well. So, and that should really be in this part of the journey here, because you can have the best therapy in the world, but if you can't prove it works, it's never going to get approved. So at the moment, we spend a lot of time focusing on optimizing the actual gene therapy and then optimizing the surgical delivery, but then we fall down a little bit at actually measuring the therapeutic effect in the clinical trials. So I went the other day and had a look on clinicaltrials.gov, which is where most of the clinical trials are registered. So they all have to be registered on a database. There are two main ones, clinicaltrials.gov and UDRA-CT. And the vast majority of them get registered on clinicaltrials.gov. Most of the pharmaceutical trials get registered on both. And all of these trials use the same vector. So that's the one that was started in Oxford. So the original trial was a phase 1/2 trial using subretinal injections, 14 injections carried out in Oxford. So I worked on this trial, and this study was conducted between 2011 to 2017 and has been completed. Then in Miami, they ran it as a phase 2 trial, used a lot of the same measures we did 6 patients, completed that in 2019. In Alberta, Canada, 6 patients, did a subset of the same test, completed that in 2021, and published that. And same in Tübingen. And we had some input into all of these, and then we did a phase 2 which looked at earlier stage patients, so patients with much earlier vision loss than many of these earlier studies, and a bigger cohort. And that's just, we're still doing the data analysis on that because that's just finished. And then the phase 3, which was run by Biogen, 170 patients, a huge trial across the world, multi-center. And then phase 2 with 60 patients, where this is still ongoing because they're doing 2 eyes. So one, and then 6 months later doing the other eye. So all of this is with Biogen involvement. Then there's the Spark Therapeutics trial, started in 2015. I don't know what's happening with that because I haven't really seen much data out of that. On clinicaltrials.gov, it seems to indicate it's still going. So, but their vector that they're using is different. And then this one is a new one by 4D Molecular Therapeutics, just starting, where they're trying to use intravitreal injections. This one was started back in 2012, but they only got 2 patients to enroll, and so they abandoned the study because they due to problems with recruitment, where they were looking at the short-term effects on night vision of taking simvastatin. And then this is a new one in the planning, so it hasn't started yet, but they're looking at vitamin A dietary supplementation, and again the impact on night vision. So that one is in Duke University in the US, and will be is not yet recruiting, but will be doing soon.
Dr. Jolly, can I ask you a quick question, please?
Yes.
A comment and a question. The comment is at the SPARC at the top bullet on this second slide. The SPARC therapeutic trial has concluded. Just make sure everyone is aware of that. And then, do you mind please repeating the second website? We're very familiar with clinicaltrial.gov, but what was the other site that you noted, please?
EUDRACT.
Can you say it again?
EUDRACT. It's the European equivalent.
Perfect. Thank you.
But the vast majority use clinicaltrials.gov. So even from Europe, most of us tend to use clinicaltrials.gov.
Okay.
But SPARC Therapeutics, I don't think they've published anything, even though they've concluded. They've been very quiet about their results.
We're waiting.
Yeah, which isn't a good sign. So yeah, I think most of us are familiar with this press release from Biogen, and a lot of people were disheartened by the fact that the outcome measures were not met. And this is why this is so important. And to be honest, I wasn't entirely surprised by this, because having worked in the trials, this doesn't mean the therapy doesn't work, because having worked in the trials, I saw the effects of it, but they didn't use the right outcome measures. And I'm going to talk quite a lot about that. And I can see that from having worked on the trials. And I think this is a problem within the field, that not enough importance is given to outcome measures research. It's traditionally just not been, it's been dismissed as not important enough, and funders haven't considered it important enough. They just want to go straight to the therapies. And I'm going to hopefully show you through today why that's not a good strategy. So their primary outcome measure, the vast majority, apart from the phase 1 where safety is the primary outcome measure, but when you start getting to phase 2 and 3, you need to have some kind of efficacy measure. And the vast majority of the trials go straight to visual acuity. It's the most used outcome measure because it's the one that everyone just automatically goes to. It's the most understood. It's what we use to clinic. It's the first thing you do when you go to clinic. And a lot of people don't like reading the charts, and yeah, it's immediately the first thing that you go to. So you're probably all familiar with the charts. What you don't realize is actually there's a lot of science in the charts, but there's also a lot of limitations in the charts. So there are quite a few different charts out there, but in the trials, we're quite specific about which ones we can use. But what we use hasn't been changed in a very long time, in decades. So the spacing, the letters we used, the progression of the letters is all very regulated to try and make it as scientific as possible. But visual acuity represents a very small amount of the retina. So this is a picture of the back of the eye. I don't know how many of your previous talks you've been shown these images, but just to recap, blood vessels here. You've got your optic nerves. So this is where all the information funnels down, and then that takes the optic nerves then connect to the brain, which then reads the information, interprets it. So that's the second half of the vision. And this is the macula. So that's where the light is focused to give you the high definition vision. And that's what visual acuity is measuring. So it's only one aspect of your vision. So visual acuity literally represents 0.5 degrees of the center of your vision. That's it. It's only telling you what's happening in this small blue zone at the center of this image. So this is a scan, the OCT scan, through a cross section through your vision. And you're literally looking through a small slice when you're reading the letters. You're ignoring all of this information about what's happening in this retina at the side. So when you have a treatment effect in any of the rest of the retina, you're not going to pick that up when you're measuring, when you're relying on that one measure. So let me demonstrate this. When you put when you're doing the operation, you're putting the virus underneath the retina. So here's an example of some cells. So each of these hexagons in this array is a single cell in the retina. So this is a small array. We inject the virus, and the green represents the cells that take up the virus. We don't want, we're never going to get 100% of the virus being taken up, because that will be a toxic effect. And I'll show you more of that later. So we're usually aiming for probably about 70% of the cells to be taken up. So you're wanting about 70% of the cells to be taken up. And in the long term, you're going to have a couple of the cells, hopefully on the edges, that are going to continue to decline, because ultimately we're wanting a bit of a stabilization effect. But if you are unlucky enough that the center cell happens to be the one that didn't take up the virus, because we don't know which of those cells are going to take up the virus, you might get a little bit of a dip here with the visual acuity, but the rest, but it stays stable. Because in the normal course of the disease, you would get a continuing decline in the visual acuity. But actually, if you keep measuring that over time, you get a stabilization. That's still a success. But on the superficial level, because you got a short decline, you might superficially think that's not a success. But actually it is, because you're not getting a continued decline over time. Does that make sense? Could someone shout out and say if that makes sense?
You know, it totally makes sense. And I'm going to try to keep the emotion in my voice to a minimum. There's some questions here too. One from Brent and another one from, there's pouring in. So Paul and Brent both have questions, but just to express the foundation, and I actually can hear Randy Wheelock's voice in my head, who's passed on. But this has been a concern of ours, and we did express this, the acuity issue, with the companies right from the beginning, and that has been very disheartening for us. So we, as an organization, to our members, just want to make sure that they know that this has been on our radar for a long time, and has been disappointing and frustrating, and even more than that. So let me get on to Paul's question. Here's Paul. He says, not having the suitable outcome measure, why has this been overlooked?
Let me get on to that. I will come to that later on in my talk.
Perfect. Okay. And then Brent, Brent asked, Dr. Jolly, at what point were you not surprised that Biogen Spark trials wouldn't be sufficient and be halted? It concerns me if it was becoming evident among researchers earlier on in the phases of clinical trials.
Again, it will become clearer as I get into my talk.
Perfect. Okay, that's it for now.
Okay, because visual acuity is represented by such a small area, there's a bit of potluck here as to which cells are then taken up. So you need to be looking at patterns. And also by trying to focus on getting improvements, especially in choroideremia, what we should be focusing on longer term and like this steadying effect, rather than preventing decline rather than looking for improvements. And I understand why the pharmaceutical companies are trying to get improvements, because it shortens the time to approval. But with choroideremia, the decline is much slower than in many other conditions. So that strategy isn't necessarily going to work. And any improvement we get is a bonus, because we might be able to rescue some cells that just before they're on the path of no return, but that's not going to be the norm. The norm is that we're stopping the degeneration rather than rescuing the cells. So this is an example from the phase 1 trial. So I took these images from the phase 1 paper, but here I apologize because some of you may not be able to see these images very clearly, but I'll try and describe them verbally as best I can. So many of you complain to me about the images where you get the really bright blue light in your eyes. And I know those images are unpleasant to take, but the reason we ask you to have those is because they are the most useful images for us as the clinical staff to look at. They give us the most information. So the images I've got here on the screen that I'm pointing to with my pointer on the left-hand side is because we can see the degeneration the best on those images. So I apologize for putting you through those images every time you come in, but we rely on those images so much. So please know that we are not putting you through that for no reason. But the white flecks you can see on those images, those are the healthy bits of retina. Those are the bits that you can see with, and you can see you've got healthy flecks of retina all over. So quite a lot of islands there, and there's a yellow outline there, which is the treatment zone. So that's where the gene therapy is treating. But because visual acuity is determined by this bit in the center, that treatment zone is nowhere near the area that the visual acuity is governed by. So there's going to be no improvement in visual acuity in that patient because it's nowhere near the area of retina where visual acuity has any bearing. In this patient, the area that controls the macula, the area that controls the visual acuity, is within the treatment zone, but it's right on the edge. So if that patient turns their head slightly when we're measuring the visual acuity, it's going to be hit and miss. And that visual acuity measure is probably going to be quite variable. And looking at variability, I did do a study where, especially because when you've got small islands of vision, sometimes it's quite hard to keep your place on the line. I don't know how many of you identify with this, but keeping your place as you're reading along the lines can be quite challenging. And even when I'm pointing along the lines, we know that can be quite challenging for some people. So we looked at using electronic charts to try and control for that. And for those of you who don't have choroideremia, as an educational video, we created this video to try and simulate that and try and educate. So this is what happened. So that was the skyline of Oxford. It's known as the City of Spires because of all the libraries, and the colleges have their domes. And this is one of our nurses in the research unit kindly being a model for me. looking through the occluder at the chart and simulating what it's like to try and navigate reading across the chart when you can only see bits of it at a time. So then we did a study, well, okay, if we bring in an electronic chart where we just display one line or one letter at a time, do we get more accurate results? And we don't necessarily, we get equivalent results, which means that in a clinical trial setting, we can switch to the electronic charts. And interestingly, we found that actually patients preferred having a whole line rather than a single letter, which surprised me. I would have thought having a single letter would be easier. But I think it's just because having a whole line is a more familiar thing, and you have a bit more feedback about where you are. So yeah, that surprised me a little bit. But then we went on further and looked at this further and looked at how does changing of the visual field affect this measurement. So one of my colleagues went a bit further with this and said, well, okay, if we think about how the visual field changes. So I've in the past, I've looked at, so this is a color picture, and the reason we can't use a color picture, it's hard to see the borders. So that's why we have to use that the image where you're bombarded with that bright blue light, because it's easier to see the borders of the edge of the retina. And then this is the different types of images we can collect, and that's the visual field test. So this is the microperimeter. So those who come in to Oxford will be very familiar with this test. And that's the simulation of the impact of the visual field. And we can map how the imaging changes and the change of the visual field happens over time. So this is the corresponding imaging in the visual field and how that changes over a few years. And by doing these studies from the patient data, we now know what are the expected changes we expect over a period of time. So with our patients, we can now advise them how they're progressing compared to the average that we see. But and from this, we can actually see the patterns of loss that we get. And this is quite important because we can see that it's not even, so it doesn't come in the same on all sides. Actually, the temporal side, so the side away from your nose on both eyes, is better preserved. And we have some theories about why this is. It may be due to how the photoreceptors are like how dense the photoreceptors are. It could be due to the underlying blood vessels. So that gives you a bit more protection. It could be due to the retinal pigment epithelium and how much protection that's giving. We have a few different theories about why this is, but we see this pattern. So this image was created as a map from 50 right eyes, 50 left eyes pushed together. So this is quite a consistent pattern we see across people. And we related that. So when you're looking at the chart, in the right eye, we know that this side on the right relates to the temporal retina. So the bit on the outside. And if we look at the number of errors we get there, so my colleague Laura Taylor in Oxford found that you get less errors on that side of the chart compared to the other side of the chart. So again, this provides support for maybe we should be switching to an electronic chart or looking at maybe other types of chart. So again, this is another limitation of focusing on visual acuity. And And then another problem with visual acuity in choroideremia specifically is it's preserved for many, many years, which is great if you've got choroideremia. But then if the trials then are focusing on improvement, if you've already got 20/20 or 6/6, as we say in the UK, vision, you can't measure an improvement in visual acuity. So we need to look for alternatives. So one option is to look at low luminance visual acuity, where we put a filter in. And in some ways, this then simulates darker vision, like darker light levels, which is where in choroideremia you have more difficulties anyway. So we're recreating the difficulties that you have. And we found actually this does pick out earlier difficulties. So it's a better predictor of who is having difficulty much earlier in the disease process. So everybody has difficulties, has lower visual acuity when you've got the filter in place, but you lose more letters with choroideremia and with other inherited retinal dystrophies compared to someone who doesn't have any retinal disease. But then at the moment, low luminance VA isn't used as an outcome measure. So then you have to start doing all these extra studies around that, because it's not just about doing the test, you need to work out how much change is significant. And this is one of the reasons why outcome measures are not used appropriately, because it's not regarded as a valid field of research, or it's not given enough importance as a field of research in its own right. What people don't realize is how much work goes into doing outcome measures research. So each time you have to look at a new test, even if it's a test that has been used before, you need to validate it. So you need to look at how much change is significant. So what is a normal amount of change? What is the order of testing? So with low luminance visual acuity, some of the trials were getting you to put the filter in first. Some of the trials were getting you to do the normal testing first and then put the filter in. But that makes a big difference, because if you don't put the filter in first, you might actually learn some of the letters since you read less with the filter in. So you might be able to cheat a bit. But when you point this out, then it's not taken on board, that can affect the results. So actually that's quite important, and that can also affect your test retest variability. Which charts do you use? As I've said, the different charts have different pitfalls. When you put the filter up with an electronic chart, it's not equivalent to using it with a traditional chart, because the luminance of the screen is different to a backlit chart. So you get different scores using the filter, and you need to adjust for that extra luminance. So you need to put in all the background work. And are we actually measuring what we think we're measuring? So we need to then validate against the imaging, against other measures, against the microperimetry. We need to understand, what are we actually measuring? Are the conditions dark enough? Do we understand what information we're actually producing? And are we, the FDA, so the Food and Drug Administration, who are one of the regulatory authorities that approve do the approvals as the final treatment, they want to know, are you improving the daily life. So when we look at the different tests, we need to understand in terms of daily life, but there's a lot of different ways of looking at things. So let me give you another example. So one of the other types of visual acuity I looked at was reading acuity.
Dr. Jolly?
So, yes.
I was waiting for a break. We have a couple questions about the chart. So maybe before okay, let's do here. This is another question from Paul. When I view this chart, why can I see to read individual letters more successfully reading from right to left?
Yeah, so that all comes back to what I was saying about the shape of the island that's preserved. So whether you're reading into, so if it depends, because your shape of the island is not even. So if you're reading into the shape that's preserved or reading out of the shape, if that makes sense. So if the shape of the island is more bulbous on one side, it's probably easier to read into that shape.
That makes sense.
Yeah, into that side.
Here's another question from Brent. In my experience, a whole line helps establish an area where you can pass your remaining vision porthole, in quotes, before and after the letters, so that any possible peripheral vision might fill in the information in the brain to complete the picture of the letters. A single letter might be hard to find when all a patient has is a tiny porthole of vision.
Yeah, so with the single letters, we don't just give you single letters, we do give a peripheral lock on it. So we have lines and a box surrounding it. Because exactly for that reason is because just a single letter without a lock isn't equivalent to a line because of the way that the brain processes that information.
He added on, I guess my comments may be described a way of cheating, my bad.
No, it's not cheating. It's absolutely right. And yet we are very aware of that effect, which is why there is a whole field of research that has looked at, it's what we call the crowding effect.
I'll have one more question here from Eric. You mentioned 70% is the targeted cells receiving vectored therapy. What evidence is there to say 70% occurred? I can't
remember if it's exactly 70%, so don't quote me on that, but that comes from the animal studies.
Okay. He continues by saying, we have read where the transduction for AAV2 vectors is not as efficient as, say, AAV8.
It depends which cells you're targeting. Okay. So the different AAVs are used for different gene therapies because it depends on which actual cells that you're targeting. So for RPGR gene therapy, for example, a different AAV will be used because you're like for choroideremia, we want to target the RPE, whereas that's not the case in the RPGR. So that's why they use different AAVs.
Just the last part of his comment here, he writes, AAV2 transduction has below 50% in treated cells in the targeted RPE cells of the targeted injection area.
Well, it also depends on the promoters used. So it's not just the AAV, but also the promoter. And that's the difference between Spark and the vector that we use is the promoter is completely different because the promoter enhances the transduction.
Okay, and you said that
but I'm not a biologist. That's not really my expertise.
But the promoters were different depending on the Spark trial and the Biogen trial.
Correct.
Interesting. Okay. Hey, can I ask one more real quick since I have you? Dirk asks, why don't measure, let me see. Why don't measure how a person can move in a dark environment before and after treatment? Oh, there you go.
Okay, interesting. Because mobility is really hard really hard to measure. And the LCA trial, I know they got, so they, interestingly, they're originally, their MLMT maze was their secondary outcome measure, and they didn't meet their primary and they got their secondary approved, which is actually really unusual for that to happen. But the MLMT is really hard to implement because it needs quite a lot of space. And in Oxford, we didn't have that space available. So, and actually one of the projects, I'm trying to recruit a PhD student at the moment. Applications are open. So if anyone is interested, please apply, because I'm trying to build a diverse lab and I would love to include everyone in my lab. So if anyone's interested in coming and doing a PhD with me, come and apply. We are hoping to try and develop another mobility measure where we can hopefully build that into future clinical trials. But it's difficult. It's a really difficult thing to do. It's complicated.
That's it for now. Why don't you continue? Thank you.
Okay, so with reading, one of the things we were able to look at with reading is how much retina do you need to be able to read? So what we found is if you can fit the entire letter within the area that you have left, you can actually identify that letter. So this is a small slither of retina, but because the entire E fits it within that in the middle image, you can read that. Eye will be able to read the retina, the E. However, this in this image on the right, the H will not be read because it doesn't fit within the island. The E next to it will be read because it would fit within that island. This is a big island, so that this eye would be able to read quite comfortably because it can see several letters. It can fit a whole word in comfortably. So this eye would be able to read quite comfortably. So when we're thinking about reading, we can look at reading speed, we can look at reading acuity, we can look at reading fluency, we can look at reading comfort. There are so many different aspects to reading. So it's not just as simple as we can measure reading. We need to identify, well, okay, what aspect of reading do we think is important? Then we need to think about, okay, well, what aspect of reading do we think is important to the patient? What do we think is realistic that we can measure? And then what tests are available to measure that? What will the FDA accept? And then can we validate those? So do we know what's repeatable? Do we know what's clinically significant change? Do we know how to reduce variability? Can we standardize those measures so that it can be measured reliably in different centers, and that we can write reliable guidance, that we can get reliable measurements back? And I don't think enough attention is given to all of that in order to get reliable data back. And that's, so anyone who knows me from Oxford, any of my patients can probably attest to how much of a stickler to detail I am. And this is what I've built my reputation on. And this is why I've then supported, like I now work with Melbourne and so many other centres. And I went and trained some people in Tubingham, and because I'm known for being that stickler in detail, and I get good quality data, which means that if you get reliable results, you can trust what you're getting out. But I'm not sure how much everyone else appreciates that attention to detail, because if you're not a visual function specialist, I don't think you necessarily understand how important all of that is. If you're more focused on just clinical trials, you don't realize how important that process is to get to that point. And that's one of the reasons why outcome measures aren't given enough importance. And the media has a role to play in this, because the media and funding bodies, just to a large degree as well, they focus so much on trials, and they just want to report the bottom line and don't give any attention to the process and how reliable the process was and how we got there and what everything means in between. That it's then hard for people who work on the process to get their point across. So I've talked a lot about visual acuity and that side of things. So I'm now going on to visual field, which I've touched on briefly. There are two types of visual field. One is the full field, so like the wider field, and one is just focusing on the central field, but looking at it in great detail. So this is an example of the full field. So there are some older tests where, so again, some of my patients in Oxford will be familiar, where we bring in the light and then I draw in the points where the light becomes visible. We get them to press the button. This is a new test I designed that actually just got published today. The paper where this is like a simple screening test, because the FDA often want us to look at the wider field as a safety measure. So actually we don't need to be spending too much time on it. So I designed this in mind that we can reduce the amount of time we spend on the peripheral field. But actually, the red points are where you can't see, and the green points are where you can see. And you can see, so this is a field plot from a choroideremia patient, and you can see there's lots of green islands there that you can retain. These two field plots are from the same person. And this is the central field on the right-hand side are a combined microperimetry plot from the two eyes. So we've created an algorithm where we've combined the two eyes together. So sets combined, so the center is well preserved, and then in the like just outside the center, the vision is reduced, but then in the far periphery, there's quite a lot that's preserved. So it's worth looking outside there in that region, because there is quite a lot preserved, but this new test takes 5 minutes and we get just a good rough estimate of what's going out there. And then we can spend more time on the center. And this is using fundus guided perimetry. So what that means is that we've got an eye tracker, so we can track the eye movements while we're doing the test. So it's more accurate than your typical visual field test, because we can, to some degree, not entirely, but to some degree, compensate for a little bit of eye movements. If someone's moving their eyes a lot, then it's not very accurate, but to a little bit of degree, we can compensate for their eye movements. But again, we need to do a lot of work to understand this. And I have literally spent 10 years understanding microperimetry. How do you get repeatable results across centers? So what are the test conditions? So when we first started doing this, because it's a much more sensitive test than your normal visual field, we started by putting people in the dark for 20 minutes to adapt them to get the more sensitive measures. And then I started thinking, well, do we really need to do that? Because actually the light we're subjecting them to is quite bright. So I did a proper study where we adapted them for different lengths of time, did the test, and then looked at how sensitive they are and realized, actually, no, we don't need to do that. If they've had imaging, we need to adapt them for 5 minutes, but otherwise we don't need to do that. So then that saves a lot of burden on the patients, and we can shorten the amount of time that you're in clinic. What is the test retest variability? Because and how much is there a learning curve to this? What is the effect of dilation on the test? Because we know that with normal visual field testing, once you've dilated your eyes, you can't do visual field tests. But microperimetry is designed in a completely different way, and the lights are being shone directly on the retina rather than in the visual space. So is that still valid? And if not, then we can change the order of the testing, which again makes things a lot easier. We needed to investigate all of these things, and each of these questions needs a different study. You can't just do it all in one, because otherwise you can't answer the question properly. And with test retest variability, you need to look at test variability at different times of day, coming back from one day to the next. If you do the tests a week apart, does that change things? If you test them twice on the same day, what are the differences? Because how do you know what is a clinically significant change if you don't know what is the change, the difference just from repeating testing? How much of a learning effect is there? How many times do we need to do the test for someone to learn to do it properly? And those are important questions, especially in a clinical trial, because until you've got reliable results, how do you know how much change you need? And the change that the FDA are asking of us might be unreasonable, because they're just basing it on a different test from a different disease from historical data. They have no background data from this test because it's too new. So maybe we can get them to change the criteria that they're governing all of this on. How should we analyze the results? At the moment, they're just looking at average of all the points. But mathematically, if you're looking at average and you're putting in all these 0 points, then you're going to lose a treatment effect if you've got a lot of non-seeing points. You know, if you've got a few points in the center that do really, really well, and then you've got quite a few points in the side that don't see, but nothing's changed there, you're going to lose your treatment effect. So then the FDA said, oh, we'll pick 5 points at random before the trial starts. But if you happen to pick 5, like if you haven't random, because you're picking at random, you can't pick and choose, those 5 random points happen to be outside the treatment zone, then again, you're not going to pick up the treatment effect. So that strategy isn't going to work. So then we looked at, well, if we do a volumetric analysis, is that going to be a more sensitive way of looking at the whole field? So we had to do a whole study around that, but then you have to then present that data and say, well, will the FDA accept that and present that to them? But ideally, all of this has to be done before the clinical trials, so you can get these things agreed. But with choroideremia, it's almost been back to front, because we've rushed into the trials without doing all of this background work. And then we need to look at what is the normal progression over time, because then we can look at what's happened in the trials and say, actually, we've slowed things down. But we can't say that unless we know what the normal progression over time, expected progression over time is. And then again, what is the test actually measuring? So, originally we thought it was a mix of rod and cone response, but actually a lot of the work I've done has shown this is more showing what the cones are doing. So it is more a reflection of daytime vision and not lower light level vision, which is what we originally thought. But after working with Maya, the company that produced this kit, we've now got Scotopic microperimetry, which allows us to do microperimetry, but at much lower light levels. So we can actually measure what's happening at nighttime conditions. So we test with the blue and then with the red light, though you don't perceive them as blue or red because the light levels are so dim, and then look at the difference, which gives us a lot more information about what's happening in low light conditions. And yes, this is awful to do because I've done it myself and it's really tough to do, but this gives us so much information, especially for patients who are earlier in the disease process. And this is really important because if we don't design clinical trials for patients who are earlier in the disease process, they're never going to get approval to have the gene therapy. So we need to have outcome measures that are suitable at that stage as well. And I started this work in Oxford. The paper's going through review at the moment. I'm working on a second paper with my colleague Laura Taylor, and I appreciate all the patients that have sat for us to do this because it takes ages and you have to sit in the dark for 45 minutes before doing this as well. So we're ever so grateful to you for doing this, and I'm trying to get funding to buy one in Cambridge for my new institute at the moment, so I can continue doing this work in my new institute. And then the idea would be that we can put the data sets together from Oxford and Cambridge, which will make it much stronger. So if anyone would like to contribute to that fund, please do let me know, because that would be really exciting work to keep moving forward. And I've got a PhD student in Australia who's looking at female carriers of choroideremia. So if anyone would like to volunteer as a patient for her, then again, please do get in touch, because she's looking for volunteers to sit for her, because we want to look specifically at what's happening in carriers as well. And then we have to do the whole process again for scotopic microperimetry, so answering all those questions about reliability, repeatability, what are we measuring, how does this relate to day-to-day life, can we validate this against other measures, questionnaires, etc. So before I talk about other measures, I'm going to talk to you about the patterns that we need to be looking for. And I'm going to pause here in case you've got any more questions.
Hey, great minds think alike. So we would really encourage you. Kathy is our executive director, and she said, please encourage her to apply for another grant. So do it. You should do that.
Well, yeah, I will. I'm going to talk to you more about, like a little bit more about this afterwards, but about funding directions.
Okay.
Yeah.
Yeah, we do have some other yes, we have some other questions. So Brent wanted to give you a big shout out. Congrats on getting published today.
Thank you. Paper number 50, most of them on choroideremia.
Wow. Here's a question Diana is asking. I think it's most likely the protocols need to be revised in order to demonstrate that one test is not necessarily suitable, while we need to demonstrate the treatment is preserving while not curing. Will that be the best approach to get the FDA approval for revision on the protocol? Are any plans to communicate this to the FDA? Timing is in a critical path to slow the vision loss.
I don't know what Biogen's plans are because I'm not involved in that process. I've answered all the questions that they had for me. I've offered my services. So yeah, I don't know what they're doing.
Okay. Hopefully we'll be able to get that from the foundation. The CRF would be able to get that information too and pass that along as soon as we do to our members. We have another question from, oh, excuse me, someone was texting me a question. Orally asks, your PhD Your PhD student is looking for volunteers in Australia only, correct?
She is going to be coming to visit me in the UK next year as well. So if anyone's willing to undergo any testing here in the UK, then as long as I can get the funding in place to get the Scotopic Maya before she comes, then we can arrange that as well.
And she also asks, how much is the grant that you need?
For the Scotopic Maya, it's about £60,000.
Okay. Please
Okay, so before I talk about other things that we can measure, I just want to talk to you about the patterns that we look for. So again, this has all come from my experience of working on the clinical trials. So, and I've worked on the trials for about 10 years, not just on choroideremia, but on some of the other RP trials as well, and not just gene therapy, but some of the other trials as well. So what I've come to realize is that the patterns are more important than actually looking at, so the patterns over time are more important than picking out specific results. So going back to this concept of not looking at how much transduction you get. I mean, obviously in human eyes, we can't directly measure that. All we can do is measure that from imaging and from visual function measures indirectly. Because obviously, unlike animal eyes, we can't sacrifice the eyes and measure that. And I wouldn't want to, because I love my patients too much. So, with underdosing the eyes, obviously you're not going to get enough transduction, so you might get a small increase initially, but then you're going to get a sharp decline over time, which is probably similar to the natural history decline that you would expect. So this is where having the natural history comes in really useful. So I've drawn a graph here where we get this maybe small increase, and then you get a curve going down, which is likely close to the natural history. If you get an overdose, I've drawn a graph here where you get a sharp increase because you get 100% transduction, which shows a sharp increase in the visual function that you're measuring, or the imaging that you're measuring, and then you get a sharp decline over time because you get a toxic effect. Because if you got too much virus, you get toxic effect on the cells and they're going to die off over time. And then what you're really aiming for is this kind of steadying effect. You might get a slight decrease in the whatever visual function that you're measuring over time, as some of the cells on the edge continue to deteriorate, which is what you'd expect because you can't get 100% transduction. But generally, you're going to get a steady state over time. And compared to the top graph, where you've got a decrease in that graph, like that line's going down. But what we've done is we've flattened the curve, and we've probably heard that a lot with COVID, flattening the curve, but that's the effect that we're aiming for. Just with visual acuity, one thing to bear in mind is this potential kink, but overall, you're still getting that stabilization. So, and that's the success that we're really looking for. And there are lots of different aspects that you can measure with vision. So you've got your detailed vision. So I've got, to represent that, I've got a picture of a leaf here, and you can see the detail on the leaf. So the veins of the leaf on there. You've got night vision, so stars in the sky. And that was the image presented to us by a patient that inspired the name Night Star, which then got bought out by Biogen. And then you've got color vision, speed of vision, which is something I've thought a lot about, but I was hoping to develop some ways of looking at that, but haven't it's been a bit complicated, so I haven't delved into that yet. And then there's contrast sensitivity. So I'm going to talk about those in turn. So night vision I've talked about with the Scotopic microperimetry. And then the other technique that's coming in, taking off now, is using this bit of kit. So, the bit of kit that I've used to do this is the ESPN. So this is normally used for electrophysiology, which is my clinical specialty. And so we can do two things with this. One is to track dark adaptation. So this is where I blind you with a white light for 5 minutes, and then map how you recover your vision as you adapt to the dark. So this is actually me, my recovery rate. And you can see how my vision gets more sensitive over time. So as the curve goes down, this is me becoming more sensitive to the dark over time, and then it stabilizes out. So over the first 20 minutes, I become quite steeply more sensitive, and then that becomes gradually more, that stabilizes out. In choroideremia, we see lots of different patterns depending on where you are in the disease process. So that process takes a lot longer. And towards the end stage of disease, you don't get down here, you just get this top bit, and then it stays stable up in this top bit of the graph. And we know the different parts of this curve. So I've done a lot of work on characterizing this, and then looking back at work that's been done in animals to understand what the different bits of this curve mean, to help us understand what that means in choroideremia. So if, depending on what curve we see, what does that mean as to what's happening in your disease process? And what does that mean in terms of what we can rescue with gene therapy? And the other thing we can do is actually just sit you in the dark for 45 minutes and then measure the end point here. So what's the absolute sensitivity? And this is really good for later stage patients, because it gives us a really good sensitivity measure of where your eye's at. So the Scotopic microperimetry is really good for early stage patients, and then this sensitivity measure is good for later stage patients. So what I'm realizing is the outcome measures we need two pathways. We need one pathway for patients who have good vision, visual acuity, and one pathway for patients who have worse visual acuity, and that way we cover everybody. And everyone I've spoken to in other centres, that's what for their natural history studies, that's what I'm getting them to now try and do, is build in these two pathways in how they're monitoring their conditions, because one size does not fit all. Okay, before I go on to colour vision, any questions on night vision?
No, but Wayne says, congratulations on your PhD, and thank you for all your hard work with the trial.
Thank you.
As a willing participant, in many, many, many different tests, how much does the stamina of the patient affect the measurements?
A lot. And Wayne has been amazing. I couldn't have done my PhD without Wayne's patience. Equally, so congratulations to Wayne too. It does. I mean, the willingness of the participants matters a lot, and the willingness of the patients to take part in all of this matters a lot. And I will talk a bit about that a bit later on as well. But it does. If someone's really tired, if someone comes to their appointment super tired, super sleepy, or drunk, then that affects our results because we get less reliable results. Yeah, that's always something to bear in mind when you go to appointments. Because a lot of this it does need engagement. A lot of what we do does need engagement from you guys. And when we plan out what order we do things in, we do take into account what needs more effort. And, you know, when I spend a lot of time planning the patient pathway, so I try and put in things that need more effort after breaks. So more effort things come in the morning, after lunch, or I build in tea breaks into my plans. And again, I'm not sure how much everyone does that, but I know I do a lot of that planning in to plan in the patient pathway and make sure I try and give people A, the best experience, but B, try and get the best results out as well. And I plan in, I plan in tea breaks, and I plan in time to just sit and chat and give time for people to ask me questions as well, so that they get something out of it as well. So yeah, I hope when people take part in all of this, it's a two-way street, because we are asking a lot of people. Okay, color vision. So this is the traditional test that's used, and this is what the FDA likes. It's called the 100 hue. It is not fit for purpose. I mean, when you've got a small field of view, you can't see many. It consists of 85 little caps that you have to sort into a rainbow. It wasn't designed for research in mind. It was designed as a clinical test to just try and figure out if you have a color vision problem. This is the map you get out of it. So again, I apologize if you can't see the results very clearly on the screen, but the dotted line in the middle is my normal group, my healthy group, sorry, and the dashed line on the outside is the choroideremia group. And from that, it doesn't give you much information. Where is the color vision problem? Because it was designed as a clinical test. And this is one of the problems, is that a lot of teams, because they don't have an academic optometrist, I mean, there aren't many of us around, but someone like me who really specializes in this stuff on the team, who can advise on this, designing the protocols, who can say, no, this isn't fit for purpose. We need to throw this out and we need to go to something a bit more scientific. Also, that test is really arduous, as those of you who've done it can attest to. So I brought in this computer test. At the moment, it's not FDA approved, and the pharma companies won't let us use it because it's quite expensive. So at the moment, it's only used in the phase 2 trial because to give us more information. But in the future, I don't know if it'll get switched. I have now got a paper copy of this, which I'm hoping to validate. But this is much simpler. It's 4 big discs and it's quite a big screen, and it's much easier to do, and it takes 5 minutes to run, and it gives us a nice output where it's 3 measures, and we spent quite a lot of time. We spent months optimizing this, where we get 3 measures, an estimate of what your your 3 cone, so your red, blue and green cones are doing, so that we can really try and hone in of what they're actually doing before and after treatment. So, and then again, from this, we were able to see that in choroideremia patients, and actually in other retinal RP patients, you tend to get more of a blue defect. And again, this was recently published information. And this was really interesting because from the general work before, this had kind of been identified, but not really. It was really, really stark with this test. And actually, when you start looking into it, it makes a lot more sense, because there's quite a lot of similarities between the S-cone and the rods compared to the other two cone types, but also it could be related to how the post-processing pathways work. So this then tells us more about the disease process, as well as being a potential outcome measure. And then we can feed this back into some of this information also back into clinical care as well. So yeah, being able to delve deeper into all of this does help us understand the disease mechanisms better, as well as giving us better outcome measures for the trials. So yeah, so it's always a balancing job between finding better tests, then getting the FDA to approve them, but it's easier to do that if we do that before the clinical trials actually start, and then finding the evidence base for them. But in Europe, we've got quite strong data protection rules. So it's not always easy just to get the data from clinics. We often have to we can only use the data from clinics if patients have consented to that data being used for research. And then catching people when they come into clinic and then getting them to consent to doing the test, it's quite a lengthy process. So all of this takes a lot of time, and it's quite a long process as well. So I think there needs to be a lot of dedicated research, which is earmarked just a lot of dedicated time and effort and outcome funding dedicated to outcome measures research specifically. Okay, so the next thing I'm going to go on to is contrast sensitivity.
Dr.
So this
Jasleen?
do you any questions on colour vision before I move on?
Do you know what? I just wanted to share that we have about 10 more minutes.
Oh, really? Okay.
We have about 10 more minutes, and I'm just going to request that the people who are attending today, if you have any questions, we have none right now, but this is the time to put them in at the last minute. So maybe take 5 more minutes and 5 minutes for questions.
Okay, so I'll try and whiz through these last few slides. So contrast sensitivity is part of like how you pick up scenes. So these are a couple of pictures from Cambridge. So I went to dinner in Stephen Hawking's college. And so this is a paving stone from the college, but you can't make out that paving stone dedicated to Stephen Hawking very well because it's very low contrast because it's the same color as the background. And then my Christmas tree you can see because it's green against the white walls, so it's a bit easier to see. So that's the concept of contrast sensitivity. And the way we measure it now is using this specialist chart, which is fading letters. But that's just a clinical chart, and it doesn't look at much of the function. So it just looks at this middle portion here. Whereas if we use gratings, we can look at this entire curve. We can look at this entire curve and get a more detailed view. And this curve tells us a lot about different parts of the visual pathway. So up here, we get more about the resolution in the eye, whereas this part of the curve tells us about how the information is processed in the brain. Then we've got different types of imaging. So this is a color image, but it's difficult to analyze this because the borders are a bit fuzzy. This is the blue image, the bright blue images that you get subjected to. It's a lot clearer, but you get different zones and different brightnesses. So we've again had to do a lot of work on how do we actually analyze this? What are the guidances we give? And if you have 2 graders, you know, how can we align those 2 graders? This is a different type of imaging with a red light, and it's looking at a different layer of the retina and giving us back different kind of information about the pathology. And then this is the white light, the blue lines that you see. And so we get lots and lots of layers, and we can scan through all those layers and pick up a volume of that, and then that's the infrared that we get at the same time. So again, the factors to consider are, what are we measuring? How do we measure that? How consistent are the measures between people? So what is the variability? What are the relationships to visual function? What does all of this tell us? How do things change over time? Then we can look at remodeling of the visual system. So from those images, so from those blue line scans, I've looked at when you get, so in choroideremia, you get these layers are affected, but actually you also get changes in the topmost layers, and that's called remodeling. So this is the thickness here, the blue is changing, and that's remodeling of the system. So the nerves are actually changing in response to the photoreceptor changes and the RPE changes. And this is what the Choroideremia Research Fund funding that has given me, and I'm mapping the visual field. So this I'm projecting into the MRI scanner, this project I'm analysing the data at the moment, and I'll talk about that in a future talk, hopefully. So that's just a sneak preview. So what does all of this mean for trials? Well, when we're designing trials, we need more visual function specialists, such as academic optometrists like myself. We need to be on the teams for determining the right outcome measures and how to analyse them, because we haven't been involved in designing of the trials at all up till now. We need more money to be dedicated to outcome measures, because most of the funding calls at the moment are all about developing the treatments. But we need to have outcome measure calls before the trials start. And we need more people to volunteer to take part in this kind of research that has no direct benefit to you guys. And I produced a podcast with Tom Walker, who has visual impairment himself and is a great media professional and great fun. And one of my previous participants got involved and got interviewed for this. And if I can ask you guys to send around the link to everybody in one of your newsletters after this, please, that would be great. And we talk about why to take part in this kind of research, because it's so important, because we cannot move this forward and do the best trials possible without this kind of research. And I am so grateful to all of you guys who have supported me in this kind of work. You are the most amazing patients to work with, and it has been an absolute pleasure, and I hope to continue working with you going forward. So thank you so much.
We absolutely will pass that along. We will absolutely do that. This has been really encouraging, and yeah, people are just writing in. Thank you so much for all of the work and all of the time that you are putting into this. The one question I had is, where do we go from here? What are the next steps? But you've outlined it in the last, oh, here they come. You outlined that in the last slide. Would there be any other things, especially perhaps with the Biogen study, that you could share with us?
I honestly don't know what's happening with Biogen. Whenever they get in touch with me, I respond to them, and I've gone through, I answer their question. I've been trying to get in touch, I've been trying to get in touch with various of the biopharmaceutical companies to offer my services as a consultant, as the visual function specialist, because I've got 10 years of experience in this arena. And it's been really hard to get taken seriously because I'm not a medic. So I just want to push the case for outcome measures, because you can see how important it is.
Very much so. Very much so.
And if more funding bodies can take this on board, and again, if the CRF can take this on board as well, that we need more outcome measures dedicated research, because that's where the challenge is. Then I think future clinical trials will have more success.
Let me continue to read these just from Amir. Thank you, Jasleen. Good to see you again. Orally says it was so instructive. Thank you. Thank you indeed, from Claire. Let's see. Paul asked, did you move to Cambridge as you felt Cambridge will be more successful in achieving the end goal of providing the gene therapy more widely to those that need it, with a smiley face? All jokes aside, I remember you making me go through that painful color testing. Your new idea looks way more user-friendly. Thanks as always to your hard work and support in Oxford. There's more.
Thank you. I moved to Cambridge because they offered me a position where I'm just going to have a lot more flexibility to do follow what I want to do. I think, yeah, I think because I'm not, so they offered me an associate professorship and the chance to lead my own group. So I can lead more of the outcome measures research. So as long as I can bring in the funding, I can pay more attention to this kind of work and just kind of really move it forward.
That would be excellent. Saranjit said, thank you. And I'm going to send you an email with some more information. Paul says, hi, Jasleen. Thank you so much for an excellent presentation. Happy to continue volunteering for research. It looks like you have quite a following. Paul asks, will there be a phase 4-5 gene therapy trial to fine tune the outcome of phase 3 successfully? This is interesting, to enable it to potentially be submitted for FDA approval. Alternatively, could the successful conclusion of phase 3, and if required, 4 and 5, be submitted in the UK for NICE approval? And if approved in UK, the gene therapy then potentially be made available in UK on NHS and or paid for privately in the UK? Or under all circumstances, must it receive FDA approval in the States before it could be considered for approval in order to be administered anywhere else?
So it needs to receive some kind of regulatory approval, whether that's FDA or EMA. So my understanding is that Biogen has been going after FDA initially. So once you've got one, it's usually easier to get the other. So whether you go for FDA first or EMA first or the other way round, I don't know. I don't know the ins and outs of why you would do which order, yeah, I don't know why they've gone for FDA first and not EMA first, but that's the order they've chosen to do it in. And I've been in touch with the FDA, so with some very talented colleagues of mine, Zisha Wu and Maximilian Val, we actually did at ARVO, the world's biggest vision conference, I think it is. Last year, we did a special interest group jointly with the FDA where we actually did a discussion around microperimetry and could we get more realistic outcome measures for microperimetry, like a more realistic agreed outcome measure. And we need to actually write that work up. But because we haven't got dedicated funding to do that, again, we're all trying to fit it in around our other projects. So it's just not, I've just not had the time to write it up. But it will get done at some point. And the whole point of that was to write a position paper where we provide guidance to researchers who are working on microperimetry that in your studies, you need to build in, the FDA advised that, like, to meet FDA approval, you need to build in this, this, and this, so that then if we meet these requirements, then we could probably get what they want changed to be more realistic.
The final question comes in. How much funding do you need for outcome measures?
Well, that's a really hard question, because the biggest thing is salary and then equipment. So, and I mean, it's quite hard to recruit clinicians, because in academia, the salaries we offer are much lower than what we can pay compared to private practice. But many of us do it for the love of our job. Like, I wouldn't trade this for the world, because I get so much love out of what I do. But if you know, I've until I got the job in Cambridge, I was literally hopping from 6-month contract to 6-month contract. And that's not conducive to being able to recruit good staff. Mm-hmm. So unless I get enough money to be able to recruit someone on at least a 2-year contract, it's difficult for me to do anything reasonable. And that's the problem.
I think that gives a lot of us from the Choroideremia, Research Foundation, we are an international foundation, and we have Canada, we have France, and we have other organizations that are in the family. And so this dialogue will really help us. And it looks like we have no other questions right now, but thank you. When we spoke, you said it was going to be very positive and uplifting and encouraging, and it certainly was.
Good, I'm glad.
It was great. And, it's late where you are, so we appreciate your time.
Oh, my pleasure. My pleasure. It's a pleasure to be here and give back to the Choroideremia community. And it's so nice to see so many familiar names in the audience as well.
You have quite a following, quite a beloved group. But when we send out the video to the CRF membership, we'll put it online. Of course, it'll be on our YouTube channel.
Yeah, so if there are any comments you want me to answer, feel free to pass on the comments, and I can send you back comments that you can then upload back into the comments on YouTube from me.
Excellent. Well, thank you so much. And for those of you that are still on, we still have about 20 people. We look forward to seeing everyone, as many as can, be at Rochester for the conference that will be held in June, the 15th through the 19th. But again, thank you, Dr. Jolly. We're just thank you so much.
My pleasure.
All right. Oh, you know what? Brent Pike, Dr. Jolly. Here's one last one, and then we'll end it. Brent usually finishes us off. I would be happy to participate in the testing. I'm 51, live in Colorado, here in the USA, already genetically confirmed. And have a high tolerance.
My travel budget would never cover flights from the US, I'm afraid. But
we'll
thank you for the offer.
That information. We'll get you guys connected one way or another.
Thank you.
All right. Thanks, everybody. Thanks for joining us. And we'll see you next time. Have a great week.
Thank you. Bye.